Okay, can you hear me? Very good. Okay, so we're going to continue yesterday's journey, right? So we're going to, again, we have no plans today. We just go where it takes. And again, I will ask questions. And I hope those students who didn't speak up yesterday will speak up, right? So, and it's okay if the answer is wrong. It's just, yeah. Oh, the Chinese name? So that's my the last name. In Chinese meaning, it says living a long life, which is a very rare last name. And like the second means knowledge or culture kind of thing. And this means, it means being different, like innovative or something like this. It's like, it's always parents' wish for their children that, you know, and who fail to live up to that expectation. So here, so yesterday we talked about this cooperatives. Engineers use the cooperation training system called Cosmo, right? And oh shoot, I somehow it shifted. The position of the bar shifted, but let's not worry about that. And we learned, right, that you know, like two mechanisms that protect the cooperation against cheaters: spatial self-organization and stress-induced, right, adaptive race. So I kind of want to tell you more about the sort of like maybe like more like entertainment, right? To tell me about my journey, right, through Cosmo, what I learned from the system, especially about the role of math in biology, right? So like then a very basic question is that can cells evolve, right? To be, because the things that we have this Cosmo, we can grow them, passage them, right? The question is that will they become more cooperative? So how would you measure that, right? Given that, like say, right, I can write down the two strains again, right, so we don't, right? So there's one's L minus A plus, the other is A minus L plus, right? So, and I want to, this is focal strain, and I'm going to focus on this one. So how would I say so I evolved them for a while, and I pull some isolate out. I say, hey, this is more cooperative, I argue. Then how would you verify that what I'm saying is true or not? Okay, this lady here, you tell me, like, what phenotypes do you expect to see if it's more cooperative? Anything, it doesn't matter, right? It just doesn't matter. It's a very small audience and it doesn't matter. And that's how you build your confidence. You say something, and if it's wrong, it's okay. It's not a tricky question, I would say it's not a tricky question. So what do you expect to see that, right, to say for this to be more cooperative? What did you say? Should be seen. What do you mean? But the both of them. So if I take, you know, like, so in experimental, as experimental, I'll play them out on a plate, and this one has a red fluorescence, for example. So now I can distinguish it from the partner. I pick some out. I can pick multiple isolates out. I want to ask whether they're becoming more cheating or more cooperative. So what kind of, you know, within the context of this system, what are the assays you will do to see whether they're becoming more cooperative or more or less cooperative? Very good, very good, very good. If it can, right, in the presence of the other, they're both thriving better. Very good, that's very good. Okay, both thriving better. I will write it down, right? So if you have a mutant, right? If L minus A plus mutant plus this A minus, and the partner we can just have it fixed, right? You know, together grows better than the ancestor. A minus L plus, right? Sorry, I didn't. So this means mutant. That's very, very good, right? Very good. So what if I say, this is a very good assay. And what if I say, I just want to look at the monoculture, right? I would I will not give you partner. Then what do you see? What do you expect to see in what kind of phenotypes? And right, so if you think about it, it's birth, death, release, and consumption, right? So out of 4, right? So what do you think will change? So like maybe you now. I'm sorry, I'm going to start using my London teaching technique now. I'm just going to point one by one, right? So what do you think will happen? So birth, death, release, consumption. Very good. Release will be more, right? So higher release. So higher release. Right, it's a higher release. Anyone has some modification to make out of higher release? This is open. It's a higher release. So if I'm say cooperating with you, I release more. Is that a sufficient condition to say I'm more cooperative? Yeah, but if you, yes, so the things that, it is true. If you release more, the other strain grows faster. So that's enough. Is that sufficient? Good. So suppose it doesn't change the fitness cost to itself. That's all very good point. Because if you release more, you are growing so crappily that your partner, like you're barely surviving, and your partner will suffer because of this. That's a very good point. So suppose fitness effect does not change, right? And then are there other things? So again, release, consumption, birth, death, right? So like a birth. Suppose birth, death don't change much, right? So let's think about consumption. If I give you a lot, but at the same time I'm eating a lot of what I'm eating, I'm eating a lot of what you give me. So then it's not actually more cooperative, right? So it's sort of like it's almost have to be release rate per consumption, right? So that is so higher release rate. Higher, like A release rate per lysine consumption. And then we'll see these two are actually equivalent. So we'll see, yeah, actually, yes.
You're saying if I am consuming more lysine, I am kind of becoming less cooperative?
No, so suppose the ancestor, I consume 1 of lysine you gave me, I give you 5 adenine. But now if I give you 6 adenine, but I'm consuming 3 of lysine, right? So then it's not as cooperative in the sense that, right, like the ratio is actually reduced, right? So exactly, yeah. Okay, so this is what I'm saying by pro-cooperative trait is like the release of adenine, the rate of adenine release divided per lysine consumption, right? So we ask what happens, right? So then this is, it's here. So we pick some mutant, so adenine release rate per lysine consumption. They're like just indistinguishable. And the reason for this large error bar is the following, right? So I have a student, Sam Hart. He is a very talented experimentalist, right? So he would do the following, like ancestor, he will set up 3 replicates in one experiment. And then he pick up some evolved clone. He will set up 3 replicates, right? So he will measure like that trait, right? So it will be something like this, right? So it would be ancestor. So it will be like this, this, right? So this is a release over consumption, right? And evolved. So in experiment 1, it will look like that. It's nice, right? Because these it means very reproducible. Evolved is being more cooperative than ancestor. But then after a couple of weeks, he'll repeat the experiment, right? So he'll repeat the experiment. And what he will see is the following, right? So something like this, right? Like this. And that would be like that. And then overall, the error bar, that leads to the very, very large error bar. Right? So we couldn't make sense out of this data, right? Because each time we control everything, like cell density, how long, because we have to starve them to do the experiment, how long we catch them exactly at the exponential phase. We do all this highly controlled experiments, but still we have very, very high variability when we do this. So we're chasing our tail, right? So I think for like maybe a year or two, just trying to control everything, trying to see why are we getting such, because the assays themselves are fine, right? Because the error bars within trial is very low. It's very tightly clustered, but the minute we go to a different week to do experiments, we get very large errors, a large difference. So trial to trial variation is very large, while intra-trial variation is very small. So we were puzzled. And I thought about it. Eventually, I realized, right? So these parameters, actually, in fact, these ways we measure phenotypes, which we talked about yesterday, they were actually not good enough to tell me the growth rate of the community. So in the following sense, right? So in the following, let me see. Also, this is Sam Hart. So like I said, we go back to this. We go back to the community growth rate. So that's what that's Sam's experiment. But I try to do the same kind of thing, same experiment while I was a postdoc. So the growth again is initially it's like the lag phase, and then they go up, right? Then they go up, sort of like exponentially, because the ratio is fixed. So the two partners grow at identical rate, and it's the same as community growth. And these are the equations. So then how would you calculate, like from these equations, how would you calculate the steady state growth rate, the slope of that, right? So, so actually, let me think, right? So I can derive it, actually that's very simple. So if we look at so if they're growing, right, because the growth of these two strains depend on the concentrations of lysine and adenine in the medium, right? And if the two strains are growing at the fixed rate, right? Because slope is constant at this point. What does that imply about the metabolite concentrations? The question is that, so the growth rate, so suppose this growth rate, right, like this looks very straight. So the slope is not changing. So that means these are not changing. So if these are not changing, and the growth rate we know, like of L minus A plus depends on the lysine concentration. So what does that say about the lysine concentration if the growth rate is not changing? It's a what? It's a what, did you say? Sorry. Yeah. So exactly, it's constant. So we can set this to 0, right? And the same for this, we can set to 0, right? So like, let's write down, right? So the release rate of A minus L plus equals, right, the consumption, right? And then the birth, I'm not going to write L anymore. L minus A plus, right? And then for the second one, it's RA L minus A plus equals CA BA, right? A minus plus, right? So then what we can do, we can multiply both sides, right? We can multiply both sides. And this cancels out, right? And that cancels out. And now you have BL and BA, but we know what's the relationship between the birth rate of L and the birth rate of A from that graph. What's the relationship? A hint, right? The ratio is fixed. So the birth rate have to be what? They have to be the same. They have to be identical. If they're not identical, the ratio will not stay the same, right? Because if there's any difference in fitness, the ratio would diverge exponentially. The fact that you can also see they're parallel here. Yeah, so let's ignore death rate. Like, yes, I forgot to say, like, the death rate is actually very small. So the net, right? So that's good. So then the same, right? So it's a community equals community growth rate, right? So I would say community growth rate of community. So then the community growth rate then becomes, right? So then this becomes So community growth rate then becomes what? Becomes what? What? This you should be able to do, right? So like the person in yellow shirt, I'm just saying, what is the community growth rate? Oh, so community growth rate. So here, so like the black is the total biomass, the total number of cells, that's black. The green is the one of the partners, and then the red is one of the partners. You can see they grow identically. So I just call it community growth rate, right? Because you can see it's equals to the red growth rate, equals to the green growth rate. And then we have this, right? We multiply. Multiply this side equals multiply this side, and we cancel terms out, right? So BL, we know these are equal to, all these are equal to community growth rate. So then what is community growth rate here from this? It should be the same, exactly. The BL equals, so if I multiply, I can just change it out, right? So I can just, so now let me just clean them up, clean this up. So then this side becomes, the left-hand side becomes RL times RA, right? Just multiplying. And the left-hand side equals CL times CA, right? And then this equals to the squared, right? So it's the community growth rate squared, right? So then this becomes this divided by CA, CL, right? And then square root, right? So that is the community growth rate, right? So it's when death is very negligible. So, and so this is actually very nice equations, right? In the sense it tells us the product of release rates divided by the product of consumption amounts per birth, and you square root it, right? So what's the release rate? What's a unit? Let's do a unit check, right? So the release rate unit is what? Somebody say something. It's what? It's moles per. Okay, so the unit. So the R is say mole. Then per time. Yeah, per time. Sorry, per time. Actually, per cell, right? And what's the consumption unit? What? Yeah, but exactly. But it's like adenine lysine, they cancel out the mole per cell, right? So we cancel this per time. And it's exactly what you expect from growth rate, right? That unit is, it should be per time, right? Per time, yeah. Okay, so now, so we try to see, right? So that is, so this is why math model, to me, that was a very important lesson, right? It tells me that you don't need to measure these parameters, right? You don't need to measure like this, how the growth rate changes with the lysine concentration, how growth rate of this changes with adenine concentration, right? And so like, because of this, you know exactly what parameters you have to measure. You have to measure two release rates and two consumption. And we covered that in the last lecture. And you don't need to measure these to understand the community growth rate. Right? So I feel, to me, that was a very important lesson, right? Like, sort of like, if you think about it quantitatively, you can save yourself energy for doing experiments. Like, I'm trained as an experimenter, still I try to minimize the number of experiments I do. And that is, math provides you with one, like one tool for doing that. Okay, so then let's go back to the, so this is what we do. So we can do replicates of this, we measure, right? So it's ancestral, like we start with ancestral cosmos. The growth rate at the steady state, like the steady state growth rate, the slope of that line, it's like that. And then I told you how I measure the release rate and consumption in yesterday's lecture. And we did that. And then we get this. In batch cultures, like in starving cultures, we get this. And even though it doesn't seem like a big difference, but you know growth is exponential. So even some small difference in growth rate would cause like exponentially divergent predictions on the dynamics of community growth, right? So I knew this was a problem, right? Even when I was a postdoc, I knew this was a problem. And, but it was, it was not clear to me, like there could be so many reasons why it won't work. So for example, like, yes, I made an approximation. It's like batch culture, maybe, like Cosmo, they're growing, like they double every 8 hours. Normal yeast doubles maybe every 2 hours. It's very, very slow. Right, but that's why approximate is starvation. Because starvation is very easy. You wash away this, you do this. But to control cells to grow exactly at 8 hours, like a very, very slow growth, it's not trivial. It's not trivial, right? Because ideally, you would need to like drop in a little bit of medium, right? And very slowly, but very frequently. So the minute you consume, you drop some in more. And then once it's consumed, you drop in some more. So then you can control them at a constant environment. So it's very hard. So that's what it is. But I was thinking, right, because the parameters of this, right? So this is what you were saying, like it's a release of, this is the phenotype where we're trying to, the other one rather. So release, right, that is the one that we're asking cooperation, like a pro-cooperation phenotype. It's exactly part of the growth, part of the community growth phenotype, right? So basically it's a product of the two cooperativity of the two strains and the square root. That's a community growth rate. That's what I was saying, the two students answer, they're equivalent in that sense. It's exactly equivalent. Am I clear? Because for cooperation, whether it's a lysine release, so adenine release over lysine consumption, is exactly part of that growth equation, right? Adenine release over lysine consumption. So we were thinking, so if my parameters are not good enough to tell me community growth rate, maybe I shouldn't trust those assays, right? So so we used it. So eventually we decided to, to actually do that experiment, right? So basically put cells not in a starved environment, but rather in an environment that mimics, like poor nutrient, not lack of nutrient, but poor nutrient. So we used this device called chemostat, which was invented by a physicist, right? By a physicist who worked on Manhattan Project, but just got disillusioned afterwards, and he shifted to biology, and he invented this. It's actually conceptually quite simple. It's a very clever device. So how it works is the following, right? So suppose I again do L minus A plus, and I drip in medium with lysine, right? I can control how fast you drip in because I can set the pump rate, whether it's a fast pumping or slow pumping, right? And then I have the cells, there's a stir bar. The bottom one is a stir bar. It stirs around to make sure it's homogeneous. And then, because there is an outlet here, so if any time it goes out, goes above this, it will just flow out, including cells. So all the waste comes out, right? And then you can see after a while, so you start with this, right? So L minus A plus in this, and you can see, right? So this is live cell density, this is dead cell density. You can see they reach steady state. Right, yes.
So if it is homogeneously mixed,
yeah.
And if I take something out, why should it be diluted? I mean, it should still be.
Any overflow.
Would usually mean concentration is changing.
Yeah, because there is one that's flowing. There's always a thing that's dripping in. So the volume will increase, right? And the minute the volume goes above this outlet, it will push out. So the volume is constant now. There's something dripping in, right? There's the pump. I didn't draw the pump. The pump that pumps these fresh medium containing this L0, this the original, like the fresh concentration in, and it's being the lysine is going to be consumed and it goes out. So let's think about the mathematical representation of this chemostat system, right? So the cell number, right? They will increase because of what and decrease because stationary, right? Because in the end it gets to steady state. That means, right, the increase equals decrease. So what's the increase here? What is increase in this system? Growth, very good. Now what's a decrease? Good, very good. You volunteered this time. Congratulations, very good. That's what I'm saying, confidence building, right? And I should do this more. And because, no, I don't, be caught on the spot is very discomforting. It's very discomforting. But once you have gone through this, then you become more confident, which is a very, very good thing. It's a very small price to pay. Steady state, right? So steady state live cell density equals birth, and the reduction is death plus dilution, right? This is so here again, so here again, we can measure death is like a, like a almost 2 logs, right? It's like below, it's very small, so we can ignore it. So just to make the class easier, right? So we can ignore it, but it doesn't matter for the next one. So if death, we ignore death, it's actually very nice, right? The dilution rate, the dilution rate, right? Because you set the pump rate yourself. You know how much medium is pumped through, so you know that. So basically you play God, right? You control how fast the cells grow by controlling the pump speed. So you play God first time, right? You get to play God second time, next, right? So that's how, okay, so let's look at the dead cell density. It's just, I'm always being very thorough. So dead cell density will increase due to the death and decrease due to dilution, right? So then the death rate equals dilution rate times dilution rate, which dead cell density, you can measure from facts. And the live cell density, you also know because you measure from facts. So once, I was just being very, very thorough here. So once you know the death rate, you plug it back in, you know the birth rate. But here, since it's so small, we're just going to ignore it, right? And what about consumption? What about the lysine in the medium? So someday, how the increase is due to what? Decrease due to what? Somebody who has not spoken. I'll try to pick girls, I think, right? So like this person, the person, sorry for putting you on the spot, right? So yes, I'm talking about you with the white bottle. So how does the lysine concentration change here? It increases due to what? Decreases due to what? Because again, the system is set up this way, right? You have this, and the medium with lysine is dripped in, because that's what the cells need, right? Drip in, and then it flows out. And of course, these cells would eat it. So then what happens to the lysine concentration in the chemostat? He should volunteer again. Okay, so you're volunteer, fine, yeah.
So it will go in cycles, like when it consumes, it goes down, and when we pump it, then it again goes up and then goes down.
That's a very good hypothesis. Do people agree with that? What?
It doesn't have to cycle necessarily, but the reduction and increase.
Yeah, it doesn't have to cycle because it's just a steady state. Initially, probably, especially if you're below the steady state, then so imagine you have very, very little, very few cells at the beginning. You drip in, right? The cells will grow really, really fast because they have a lot. But once you're getting into that state, it's not changing anymore, right? So then I would say, oops, I was just being, so, oh yeah, so this is to tell you that, sorry, I forgot about the order of the slide exactly. So that tells you like so the adenine concentration we see also reaches steady state, the release. But that's actually what do we see. The lysine concentration, it's not oscillation, it's just going up and coming down. It is not steady. What do you think is happening? What is a hypothesis? What's possible? Like why is it that is steady state? Sorry, right, that's a steady state. It is a cell density is a steady state. You would expect a steady state, but it's not. And what could be the reason for that? Yeah, you speak too much, so I'm not going to. Yeah, and what about the girl in blue shirt? Do you want to try? Like, what happens to let's say, the lysine, like, what do you think could be happening? Why is it not? Or you can ask, the next question is sort of like, what determines the adenine concentration, right? What is increasing and what is decreasing? You can choose whatever problem you want to solve. So let's think about adenine concentration. What increases? What makes it increase, the concentration increase? if I put L minus A plus cells in the chemostat. Release, very good. And what makes it decrease? Because remember, you put it in and the pump gets pumped out. It's dilution. Very good. See, got it. Very good, very good. And okay, let's get to this problem then. Why does the lysine concentration not, right? It should, but it actually decreased to below the original. It was steady state for a little while, right? And then it decreased. Why that's the hypothesis? Yeah, you can speak now.
No, it might be because the cells themselves are seeing that there is lysine around, so they don't have to make it on their own anymore. So they're using the minimal lysine which is coming in rather than spending their own costs on it.
But they will never spend any cost on it anyway, because the cells themselves will never be able to synthesize lysine from the beginning. Evolving, exactly, evolution, right? So let's check whether that's the case. So for ancestor, we put them into the very low lysine concentration, 1.5 micromolar. You can see the dots, right? So each is like a cell. Each is one cell. This is budding cell. This is cell, right? But if I take a later culture, I put them on this medium, I see this, right? So for this snapshot, so like it's like a, it is sort of like a, I take a different colony. So I basically take different colonies and grow them. So for this colony, there are variants, right? So like some of the cells just don't, and some so I call it mildly evolved. And that is from a clone that all of them grow really strongly. So these are strongly adapted, right? And so we can count the number of these evolved cells just from phenotype, right? So then this, you can see this quickly increasing. So in fact, in chemostats, evolved grows like 4 times faster than the ancestor. So giving rise to extremely rapid taking over of these of this. So then what do? That we cut it off, we chop it off at here, right? We chop it off at here. So that's the tricky part of doing chemostat experiments, right? It's one time you want to get to steady state quickly, but then you cannot waste too much time, because if you waste too much time, if you get this slowly, the evolution is going to kill you. Right? So you have to do the experiment just right, so then you have enough data to do that. And it's, it's not a trivial experiment, but Sam was able to do these experiments. So we go back to the math at steady state. So consumption, so what it's very similar to what's said, right? So consumption of lysine, right? So it's like the increase is due to the dilution rate of the fresh medium outside, and decrease due to consumption and the dilution, right? And the consumption is sort of like how many new births there are. Each birth would, would reduce this by some consumption, right? And then we can do, so basically if the birth rate is the same as the dilution rate, right? Because we're ignoring the death. So the birth dilution can cancel out. And then the consumption, and this is very small, so we can ignore that as well. So just L0 divided by live cell density. So you know how much you made in the medium. You can count the number of cells, the live cells in the chemostat, right? So you know the consumption. So you play God second time by controlling the L0. Right? If you put more L0, there'll be more live cells. So you can control, in chemostat, you can control 2 things. One, you control growth rate by controlling the pump speed. You control the cell density, right, by controlling the initial, there's L0. Right? So it's a very clever device. And so then if you do that, right, and so then the release, let's go to the release. Release, right? So it's live cells, they release at some rate, and it decrease, right? Decrease due to dilution. And then the release rate becomes dilution rate, which you know, because you set a pump, times the adenine concentration at the steady state, which you can measure, because we measured here, like it's 15 micromolar, divided by live cell density, which you also know because you measured from facts, right? So then all these can be measured. Oh, yeah, yes. Hold on for a second. What did you say? Mm-hmm. Yes. Oh, it's coming in at a dilution rate, right? Because so dilution rate is sort of like a milliliter per hour, right? Your pump divided by the culture volume. From what equation? Yeah, from the top equation, right? L0 times dilution. You're asking about that? Yeah, we get consumption rate. Sorry, consumption amount per cell. Okay, so let me see. So it's here. So thank you for asking questions, right? That I'm very grateful. So when you pump in, it's coming in at L0 times dilution because the faster you dilute, the more it goes in. And so it decreases because of the birth, each birth, right? So this birth rate, so birth rate times alive, each birth would consume that much. And the plus, so the dilution is also they're diluting out. So then here, so the dilution rate equals birth rate. Because we just talked about how the birth minus death, because death is so small. Yeah, so birth, yeah, I should have put everything here, right? So basically, if we go back, this is actually good. Okay, here, right? So basically, death is very small, so we ignore it. So basically birth, right, from the top, birth equals death plus dilution. So death is small, so birth equals dilution. Let's say birth approximately dilution. Like rate, right? I'm just not going to say the rate. Right, and then, right, so then now you have this. So then the birth equals dilution, you cancel out all these, right? And then L, so L is the medium, it's a concentration of lysine in this. It's very, very low. It's like, because with a very slow growth rate, we can make them like grow really slowly. So the L is nearly negligible. It's sort of like, it's very hard to measure, right? So it's like barely detectable. So we cancel that, we just ignore that. Then you just have consumption, right? So like, let me erase some. Right, so the consumption equals, right, like whatever is in the reservoir divided by live cell density. Right, so that is exactly, that's very good. And then I will do release. So it's like this, release basically is, right, so release rate is calculated that way. Good. So when we look at the release rate, so like just all parameters, this is just one example, right? So what do we find is the blue dotted line is like the detection limit, below which we will not be able to detect anymore. So if they're growing exponentially, like 0.45 per hour kind of ish, there's actually no release, even though they are engineered to overproduce. They don't overproduce. And the starvation, that's what we did before, right? It's there. And so the Cosmo relevant environment is here. You see this. So it's like the parameter itself, right? It's not constant. It actually changes with how fast cells grow. Right. So then, and then once we incorporate this chemostat, so in one chemostat experiment, we know everything, right? We know the release rate, we know the consumption amount, right? And the birth, of course, is controlled by the chemostat, by the pump. So once we do this, we recover, right? We get quantitative agreement with experiments. So now we are confident, right? So like we basically rescue the paper. Because the paper I always want to write, but never got a chance because you can't tell, it's hard to tell students, hey, this experiment didn't work. Can you figure out why, right? So that's very good. And then when we do the experiment, now this problem went away. This irreproducibility or high noise problem went away, become really reproducible. So this, like this mutation ECM21 shows up, right? So it basically, its release rate per consumption is significantly more and reproducibly more than ancestor. It's about 1.6-fold, right? And then since we know this is linked to this, we should predict the community growth rate will be square root more of 1.6 more. And so indeed, the steady state growth rate of the partner and of itself and of the community, right? It's it is quantitatively that much more, square root of 1.6. So basically, you can't evolve, So ECM21, if you remember, I don't expect you to remember, but yesterday I talked about these cells are very good at like stabilizing lysine permease on the surface. So they are very good, they are self-serving, right? The mutation is self-serving in the sense that they can grow much better on the limited lysine in competing with other for lysine. But here we also showed that just through pleiotropy, now we don't quite understand why, right? They also release adenine more. So it's sort of like this has no evolutionary history. But this kind of win-win phenotypes can arise somehow relatively easily because of pleiotropy, because a gene can affect many phenotypes. Yeah.
After you observe that after some generation, this trend is evolving to be more cooperative.
Yeah.
In this sense,
For this particular one. But it's not necessarily true.
so for this trait that has become more pro-cooperative, did you genotype and see that there are no new mutations?
No, yeah, no new, because we can do the strict knockout. We can recreate the mutation. It's a very good question. We can recreate the mutation by knocking out this gene called ECM21.
And that itself is sufficient to create.
That's right. It's insufficient. Exactly, it's sufficient. But if we do something else, like say for example, like a chromosome duplication, so that they duplicate the lysine permease, it does not show this phenotype. So it's not necessarily true, right? It can happen. I'm just saying it can happen. I don't know how often it happens. I'm not looking at many isolates. It'll be interesting to see how often this kind of pleiotropy effects. Now, I'm sure there are ones that will decrease. Right? If you screen enough, that can either be increased or decreased or not changing, right? But I just don't know the distribution of these phenotypes. I cannot see anything special. I'm sure it has special thing, but I cannot see, or we have not invested enough effort to know what makes this so special, right? And we have even a more bizarre one, right? The RSP5, right? It's also very, very good at taking up. They actually do not over-release more, but they make the community grow faster. So then I think they're probably releasing something else, but I don't know what that something else is, right? Because the biology, that's the thing. Once you get into it, it creates a plethora of mysteries, right? And you have to decide what to go after, what not go after. But that's what's also makes it very interesting, right? So I believe this, we can have a short break and I'll switch to the next, like a short 5-minute break. So I will switch to the next one. Any questions? Oh, wow, automatic. Just somebody's controlling that for me. Seamless transition. So I guess we won't have a break because the transition is so smooth. So next I want to switch gears to some more theoretical work. It's very related to what Sylvia has talked about and will talk about. And so the question is that, but actually I want to thank the people who did the work first, Li Xie, a former postdoc in the lab, and Alex Yuan, a former graduate student in the lab. Right? And Alex also did other work on sort of like a causal inference, like measuring dependence between time series and so on and so forth, right? So I want to start with a question, right? So suppose you have some, suppose you have some waste, right? And you want to degrade that waste, right? And you go to, like some river, right? And this happen to be like pond water. You isolate some microbes. You know that this mixture of microbes can degrade that waste. But the activity is very low. How would you try to improve it such that it would degrade this waste faster or more thoroughly? How would you do that? Okay, this lady. Yes. What did you say? Oh, the question is, suppose I have some waste I want to degrade, right? And I know some, like what's the biggest river in India? I should know the answer, but I don't. Ganges, right? Ganges River, like that. Okay, you go to River Ganges, right? You find that the microbes there are a lot of microbes in the river, right? So that if you take the microbes, you put them together with the waste, it gets degraded, but it's slowly, right? And you wanted much faster degradation of this, right? So what will you do? What's the next step you will do? Increase the microbial what? Yeah, so suppose you try, that's a very good answer. If you put more biomass in, it would do better, right? But I kind of want to change that's not quite density dependent in the sense that, suppose I say fix, or suppose even if I put a lot of cells in, it's still quite slow. Then what's the next step? Yeah, very good. You change the composition of microbes, right? But that's a very good question. That's a very good possibility. So there are like ways of doing that, right? So one is sort of like trying to figure out, right? Like what are the species compositions? So first of all, you ask whether a single species can do it. If a single species can do it, just do that single species. You can like select for, sort of like that gets back to like a lot of topics, covered in the previous talks, right? So it depends on whether the degradation is costly to the individuals or not, right? If they're just secreting some byproduct, and the byproduct can chop it off, or if they can use the waste. Ideally, they use the waste as carbon source. So the more they degrade the waste, the faster they grow, right? So then this would be very, very nice. So suppose that's not the case, right? So suppose that this one species is not enough, right? You actually require like multiple species to work together to degrade more, right? So then what are the possibilities? You require 2 or more species to, like maybe one would degrade like this into intermediates, and the other would degrade intermediates to the final, right? Like the clean, the small products that are not harmful anymore. So suppose you figure it out, you need at least, right, you need 3 species, like say even 2 species in the community. They need to be together to do the degradation. Still kind of slow. Then what do you do? What did you say? That's right. That's multiple generations, right? In multiple generations, you're trying to you try to evolve perhaps better communities that can do that. So the one possibility is you will mutagenize each of them and do combinations, right? To see what mutation combinations you can do. So that is one possibility. That's called so-called bottom up, right? So you modify each, because you don't say, so this is sort of like the community, like say function, right? So it's a function that relies on members of this. So single members cannot do that. You have to have together, right? So interactions between them would generate that function. And you can modify it, but generally speaking, the interactions are very complex, right? Each microbe can release hundreds, if not thousands of compounds, right? And then many of these will affect the partner in many, many different ways. So it's very hard to map out exactly what happens. So you can do, you can do bottom up, so-called bottom. You start with the composition, the member species, you mutagenize them, and you try out, you screen through these combinations and see which one would, which community would do the best. That's certainly possible. The other possibility then is to, as you said, to do a selection, right? Selection, sort of like, like community level selection, right? But if you think about sort of like how might we improve community function, right, through selection. So then a selection cycle, right? A selection cycle, I would just say, so one possibility, there are many ways of doing that, but the one possibility is the following way. You start with a newborn communities with small, low cell densities, right? So I will mark the 2 shapes, right? The oval and the round as 2 different species. And they differ in their phenotypes. The shade means they have different phenotypes, mutants. And you let them mature. And as Sylvia said, this is artificially imposed, the life cycle, right? So you as experimenters decide how long they're supposed to grow, right? So during this maturation, the cell densities increase, the cells intact, and they generate this purple pigment. So suppose, right? And then we want the purplest community, where we want the purple pigment. And then we look at at this point, we ask, well, which community is the most purple? I say, this one is the most purple. So we'll take this, and reproduce. There are different ways of reproducing, but for this one, we just say, like we just randomly sample from this parent community, right, into different offsprings. And then the lower functioning adults will have fewer, offspring, just like what Sylvia said, is a differential fitness, right? And then the cycle gets repeated, right? And as we heard in Sylvia's lecture, like so this repeat. And then what's, so the essential ingredients of this, right, is there's a variation, right? There's inheritance, and there's differential reproduction. So I want to actually think through this, like step back, think about the selection of individuals first, right? And go through these. So suppose I have a cell that it's gray shaded, it reproduces at some rate. We have a mutant, the white one, that does not make the gray pigment, but reproduce faster because it costs some energy to make this pigment. And we could also have the opposite type, right? That grows slower, but make more pigment. Right? So suppose we want that pigment. We will grow them up. We'll say, well, this is what we'll do. We have a mixture. We'll just select, we see, hey, which one is the darkest? We select that one and only let that grow, right? And then we repeat this process. Eventually, we get cells that are darker but grow slower, right? This is artificial selection. The selection is done by us, by the experimentalists. But then you can also do this, just grow them up and not do anything. Just let the fastest cells take over. In this case, which one would be favored in this case? The white, very good. White would increase in frequency, right? So natural selection will always favor this, right? So you see this conflict here, right? So one would favor faster growers, the other one wants to favor the slow growers. And again, we have 3 conditions, variation. So what introduces variation here? Mutation is very good. And the selection, in the first case, the selection is done by experimentalists. In second, it's a natural selection, right? And the third is heredity. So if you are dark, if your parent is dark, your offspring tends to be dark as well because it's genetically controlled, right? And so basically you correlate parents' trait against offspring trait, right? And if they are correlated, then there is inheritance. So now let's think about this for community. What think through each criteria and see whether they fulfill this. So I want some students to speak up. The people who have not spoken. It'd be great if you volunteer, right? Because otherwise I have to point at you, which is even more pressure. The person with blue shirt. Right. So tell me variation, selection, and heredity. So pick one of these. For this, which one is satisfied? What's the difference between community selection versus individual selection? Can you speak louder? Repeat the question. So if I do community, so individual level selection, we have these, right? We saw the example. But so now I ask you to consider the community selection. So selection on the unit of community. So then would the variation, would there still be variation? Would there still be selection? Would there still be heredity? Are they affected? Because the unit of selection now becomes a community of cells rather than a single cell. You can choose any one of the 3, right? So and the rest be prepared, right? Because then of course, whatever she didn't pick, then probably you will answer. Selection, yes? Did it change from the individual selection versus community selection? I'm trying to figure out the difference between these 2 levels, 2 types of experiments, right? Yeah, exactly. We want to select for the purple shade, right? It's a purple shade. But except the difference is the purple shade is created by the 2 species together. It's through mysterious interactions. They make out the purple pigment. So I'm selecting for the purple versus in the previous slide, I'm selecting for the dark cell, right? It's all artificial selection, right? I already gave away the answer, right? The dark pigment but one cell And this one is a purple pigment made by two different types, two different species. So I want to go through these three criteria, because to be successful, all three have to be fulfilled. Right? So I'm just checking whether, how these are. So let's start with selection. Is the selection here similar to the selection back there? That's right, that's right. That's right. Yeah, yeah, so it's good. So this one and the previous artificial selection are the same in the sense they are both done by experimentalists, right? That's good. And what about variation, someone else?
In differing peptides or species have differing color outputs, so there exists variation.
What did you say?
So you have, if I understand correctly, the pale purple and yellow put together produces a different shade.
Oh, the shade means mutation. They can either affect the interactions more or less, but you are on the right track, right? So variation is also here because you have mutations. Different mutations would affect this, would influence the purple, the production of the purple, different ways. So you can have, if different communities sample different mutations, then you will have, variations, right? So what about heredity or inheritance? Yeah.
But you also have variation just because of differences in abundances.
That's right, that's right, that's right. But that's coming up, right? So that's right. And the inheritance.
Hereditary means it creates more adaptability to the existence.
What did you say?
The hereditary.
Yeah, inheritance is like a similarity between parent and offspring, right? So if the parent is purple, the offspring being purple, right? So is that? I actually didn't hear what you said.
That's the preferred for the.
You prefer for both, but is that realized here? In the individual selection, there is the genome, right? Like sort of like if it's a unicellular organism, asexual organism, right? Whatever genome your parent has, your offspring generally have that with small changes due to rare mutations. But here, the parent, if you look at the parent community and offspring communities, do you expect them to be very similar, or they can be very different? So they're all well mixed, yes. And no clumping. It's not like trivial, right? It's not trivial, but it's not terribly hard either. It should be. That's a very good, it's very reasonable. It's a very reasonable thought, right? So it would be similar. So you argue they'll be similar. And anybody disagrees with that? Very good. If you're doing sampling, right? So suppose the parent has a rare genotype, right? When you partition this, the rare genotype may not be partitioned into the offspring. It could just be lost. If you're just you're just having this produce 3 offsprings, right? And you pipette out 3 little portions, it may not include that. So the parent and offspring may not be, right? May not show very strong, like as strong at least inheritance as heredity as offspring, right? So okay, so now we have this, right? So we want to, actually I would back out. So people have tried this kind of experiments, right? So most of the experiments actually turns out what happened. So if you take complex communities, you try to treat them to degrade waste, like some waste or some cellulose or something like that. In general, what happened is that it's a very quick improvement, right? That's because of the assortment, like sort of like the thinning down to useful species, to like weed out these bad species. But once then they quickly plateau, and then sort of like it just stops, and then just really, really slowly, right? Because you have exhausted variations by that point. Because initially maybe you're weeding out the bad species, but once you have weeded them out, you have weeded them out, right? And then basically and then there are also experiments that even though you try to improve the community function, it declines over time, right? Despite your desire to improve it, it went against you because you were trying to go against the natural selection, right? So then we thought, well, it was not true because when Lee came to my lab, she was an experimental physicist. So when it came to my lab, and at that time I would only admit students who do experiments. At that time, I just didn't think, I think, well, math is kind of useful, but sort of like this is experimental lab, right? And then, so I will only recruit students who will do experiments. And so she's experimental physicist, so she passed the bar. But then we wanted to do experiment. We thought, wow, it's really hard. Looking at the previous literature, it looked really hard. And I kind of don't know because these experiments, right, as some of you mentioned yesterday, the biologists will just put observation there. And it's actually really hard to, for these problems, very hard to know why, how things happen. So I said, oh, fine. So then we will do simulations, right? Because being trained as a physicist, she can also do simulations. I said, well, let's start with simulations just to see how hard the experiment is. So we try to think of the simplest possible case, right? To see how easily it will fail or it would work. So that was a motivation for the next one, for the setup. So this is completely simulation now. So two species community that converts waste and resource into product. And then you can imagine, right? So there's this helper, I call H, manufacturer, I call M. And it's some shared resource, right? Because they all need, say, nitrogen source for them. They will compete for that. And then helper will degrade waste, like say cellulose, something you don't need, but it's in excess, so it doesn't get into the math equations. And then helper would grow, right, and release some byproduct, like acetate, right? And this we see in microbes a lot. So the byproducts are at no cost to the producers, because it's just like the waste product. But this byproduct happens to be like very important for the M. The M, say M's carbon source, right? So the manufacturer will use B and R together, right, to either to partition part of the resource to make the biomass of itself, and then the rest to make the product, the purple pigment you want. And I call this FP for fraction of growth used to make this product. So it's a cost. It's a cost, right? That's the thing we're going to focus on primarily for the rest of the lecture. And then the growth, of course, is 1 minus FP, right? And I wonder whether I should, what time? I still have some time. So I want to actually, let me see. So the cycle consists of this, right? So you have newborn of small number of cells of both species. You grow again for some maturation time T, you get to adults. And the community function is like this, right? And then you do this procedure and you select the darkest communities in your partition. Actually, I still think I should write down the equations, right? So just for this, so that we have. So let's see, the helper population size changes because of what? How does that population size of H change? Yeah, but the waste is excess, so we don't consider that, right? And then it requires the shared resource, right? So the growth rate, right, which is the function of H, which is the function of the resource, minus death rate. And what about the manufacturer? Yeah, growth, but the growth, a little bit more detail. Oh, very good, you volunteered, that's really great, right? So the growth rate of manufacture is a resource and the byproduct, right? So we can also, of course, write down this, death rate, manufacture, growth rate. But there is a catch there, right? The growth rate had to be discounted, right? 1 minus FP, they're very good. Very good. Okay, so then the product, that's what we're interested in, right? The product over dt equals what? little murmur. I cannot hear. Oh, GM, I forgot M. Sorry. Oops. GH. Sorry, yeah. What's the product? Not so many people I cannot hear. So what? Yes, FP, exactly, exactly. So I'm just going to say GM, so GM, right? RB, exactly. And then the finally, the byproduct. Right, byproduct is like, it's made by some production, right? And I just normalize, it doesn't really matter. Minus consumption, right? By the growth of M, some consumption and some, some release, which scales with that. Or then of course, then finally the D resource. DP is minus consumption of resource by M, GMM, right? So this is not discounted anymore, right? Because this is just the resource required for making both. RH, GH over H. Let me just double check that looks right. Yes, right. So these are the five equations, the 5 state variables, and how they change with time. Okay. Yeah, resources like nitrogen, like they need nitrogen and they don't produce nitrogen exactly. But that's a very good question. We're getting there. Yes, yes. So exactly, there's a oh, it's a resource, a byproduct, right? The byproduct, it's depending only because of the byproduct. That's right, that's right. And these ones are just like, so this we model like this, right? It's sort of like saturable growth, right? So it's like sort of like if you have like a resource, initially if it's a 0 resource, it doesn't grow, for example, right? It's a growth rate. Growth rate of H is a function of R, and then to the maximum, right? And then the half maximal is K, right? The K, so it's like, so the growth rate, right, as a function of R equals, this is max, G max, right? It's a G max, it's GH, right? So I'm going to just get rid of H, it's cumbersome. G max R divided by K. It's called the Monod constant, Km plus R, right? So basically, this Km is a concentration of R such that the growth rate is half G max. So sort of like saturable, which is observed a lot in microbial literature. So then if you think about experiments, right, there are many variables. Oh yes, you have another question. D what? dP by over dt, that's right. Yeah. M is not, M is the population size of M. Yes, it is a function, right? In a sense, it will grow according to that. But it's the growth, the total growth. I didn't bother to write this whole thing down, but it's a total amount of growth potential diverted to make this versus biomass. This is biomass, and this is making the product. Yeah. If it didn't make this, it would have gone here, but it's. So like for experiments, so that's a problem, right, with selection. That's because there's so many experimental variables, right? And it's not clear how these variables will affect the efficacy of selection, right? So let's free discussion again, right? how do you think those experimental variables will affect, right, the three criterion, right, the variation, the inheritance, and the selection, right? Selection is like done artificially, but that is like the three requirements. And how would you go about choosing values for these? Yes, you can. Yes. You're so enthusiastic, so it's hard to say no. Yeah, go on. Yes, choose one.
Priorities of what you want to do. You want to be able to evolve for a while, so you don't want variation to die out very fast.
Yeah.
And you still want to, increase your, basically increase what you're selecting for. So you need medium, which isn't, very low, because then your selection very fast, and then you will in fact end up with a decline as your variation drops off. So you want reasonable amount of medium around, and then you would want to say your mutation rate has to be not too high, but also, again, you have to pick these Goldilocks.
Very good. Just let's focus on mutation rate, but more detailed, right? More sort of like mutation rate. You said you don't want to be too high, nor want to be too low. And why is that? Can you elaborate?
I don't want mutation rate to be too high, because then it's basically going to lead to more bad mutations and more bad communities.
Very, very good. Yes, yes. But you don't want to be too low because
then it's not doing it fast enough.
that's right. That's right. It's such a good analysis. That's very good. Okay, so anybody else, right? With adding to other aspects. So the mutation rate is done, right? It's done. It's exactly right. And then any student who has not spoken, would you like to volunteer? It's a school, right? This is a school, so. What about the students who are sitting over there? And it's okay to be wrong, right? Again, just sort of like intuition, right? And of course, intuition can be, wrong. That's why we do experiments, we do simulations, right? So I'm just curious what your intuition is, like, because the mutation rate is very good analysis, right? By the student. Sorry, I don't remember your name. So start with number of communities, right? Sort of like sort of like number of communities. So if you increase number of communities, does that help with experiments or not? And the rest you think about other ones, right? Because I'm going to call you at some point, right? So you think about other aspects, right? So think about the extreme case. If I just have one community, is that good enough? Then I cannot even do selection, right? It's just one, right? And then so if I increase number of communities, then what would it be? Then what might happen? Do you think it's bad or good? And if maybe it's good and bad, then what is good, what is bad? Anyone else on other aspects? How they think about this? Yes.
For specific choice, we need to choose spaces which will cooperate each other, not kill each other.
That's right. That's very good. So that's, and then also they will coexist, right? Exactly, very good. So like the second one is knocked out. And so when you're ready, let me know, right? And then, I just want to break the silence of students, right? So I just want to push them to, to be more like, I don't know, vocal. How about you? The aspect, exactly. So yeah, the yellow bottle, sorry. Which aspect are you thinking? Oh, number of communities, so you're free now, yes. You what? Number of newborns, that's very good. Like a number, so like we have the total amount of fixed, for example. Like of course you can change the number of communities, but if I were to say do it with a robot, I kind of want to program it in advance, right? How many, so like a fixed total number of communities, right? But it's a fixed total amount of communities, like we know it cannot just be one, right? Because then you cannot do selection. But if you have, if you have more communities, what's the good and the bad associated with it? Oh, okay, good. So my fault, I didn't make it clear. So these are numbers. So here, right, 1, 2, 3, so that's the phone number 10. And here would reproduce exactly 10, right? So like you can, of course, doesn't have to be 10, can be 100, it can be 1,000, right? It can be 1,000,000, right? Like sort of like if I increase the number of communities, what happens? With regard to the variation and so on and so forth. I see. So the hope is that, so I didn't say that, right? Like the things that the hope is that, right? When you put the cells in, there'll be some mutations, right? So if you have very few to select from, you don't have a lot of variations among communities, right? But you have many, 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 it just experimentally become very hard, very infeasible. Does that make sense? So we know like one community is not enough, right? Because it's not even community selection anymore, right? And if you have like several, like so maybe this mutation, the first one will sample this blue one. The second one might sample different mutation, like say in a partner. The third one might sample yet another, mutation. So like you increase the odds of achieving some extreme value, right? The extreme purpleness, right? So just logic. I'm not asking for quantitative kind of thing, just like logically, sort of like intuitively, right? It can't be worse, right? If I have more communities, it cannot, like it's hard for me to imagine it's worse. Because you're selecting the best. The best among the 1,000 must be at least as good as the best of the 10. Right, so that is, right. But actually like multiple people's work, including Sylvia's work, right, showed that the number of communities actually does not matter as much as you think. So this is like good for experimentalists, right? Because it's actually very hard to deal with that. And what about the medium? We talked a little bit about this. So we want to have enough resource, right? So that they can kind of grow. And what about, like, what are the things we didn't cover? Oh, the number of cells in newborn and the maturation time. Yes, exactly. That's the impose the life cycle, right? Yes. Yeah. Yeah. And I think you have to speak slower. I cannot quite hear. Yes. Yes. You are asking me a question, right? Is that you ask me? So the question is somebody can reiterate that question because it was just hard for me to hear. Yes. That's right.
And then they start making the purple product, or is it that?
Oh, I see. No, throughout. That's good. Yeah, it's throughout. Throughout the process, they're interacting. Yes.
Okay, then more maturation time will help me to reduce more waste, something like that.
Yeah, and then you, yeah.
I think more maturation time would be good because after maturation they will reproduce, so maturation time should be shorter.
Should be shorter. Yeah,
Because
and
you
then
want more reproduction so that your experiment is faster.
it's faster. And then what do you say?
I'm saying should be fast, like higher or lower, but there are different reasons for why it should be high, why it should be low. If it's lower, you get to do it faster, you get more generations within your experiment period. But if it's very short, still there are two communities that differ in purple production rate by very small amount. If I give them less maturation time, I won't be able to know. Like when I look at that, I think they're both same.
Yeah, that's right.
But
It's
if
too,
I
it's
get more resolution if I have higher maturation time.
right. But why not longer, right? Why not?
Experiment get delayed. Like
What did you say?
feasibility, like if
Oh, feasibility. So suppose it's feasible, right? As experimenters, I want to be as long as possible. That means it's reduce my workload. I don't have to go in and propagate it, right? So like, but can I just make it very long? And have more resource, because of course, if the resource depleted, you don't do much more, right? But say have more resource, just wait for a long time. But for experimenters, it's fine. Like it's fine if I go to lab, like if you know, in fact, the fast propagation is very hard on experimenters, right? If you, suppose you have to go in every 8 hours, what does that mean? That means you won't get a lot of sleep, right? So like we want, right? It's actually not right. So so that is, I know it's very interesting to hear, hear you guys give answers. But the problem is the following, right? Once you have, okay, so let's go through this, right? So this we have done, mutation rate, we use that, so it's not important. Also mutation, so mutation rate is like very high in the simulations. So 50% of mutations would set the cost to be zero, so be just non-producers, right? And the other 50% will increase or decrease the cost by a few percent. That's like experimental observations, right? And then we did this, we did the medium, right? The medium, we just avoided saturation. And the species choice, we said that they have to coexist. Oh, but I was ahead of myself. But now the order is a bit, that's a problem with free discussion, but the order is not quite, like I can do that. Species choice about coexistence, I have to have. So the helper can just grow, right? So we're going to back to the number of cells and the maturation time. Because it has, basically it just, if the resources are in abundance, right, it just exponentially grows just fine, it has the resource. But for manufacture, it can't, right? It had to wait for the byproduct to accumulate. It had to do this. So the manufacturer had to be faster than helper at some point to be able to coexist. If it always grows slower than helper, it would just be, it just goes extinct. So that's a choice, right? So then if, M pays high cost, then it just, for example, if it pays one cost, it doesn't grow at all, then of course it will always go extinct, and that's not good, right? And if it pays low kind of cost, such that it will catch up like this, then it will coexist, right? Because it will catch up at this point. And if the loads, so that is the moderate ratio we want, right? So here we will fix everything else, right? We fix everything else, just consider that one, the cost. So what's optimal? So like what's optimal cost, right? If the cost is very high, then what happens? Yeah, it just goes extinct, right? If cost is very low, it's not good, right? So because it just doesn't produce anything. So basically it's intermediate, right? You want intermediate. The goal is to do that, right? So the answers, I don't understand why the slides are not showing up. But maybe I'll go back. I still want to, I'm obsessed with that, with that cell number in the newborn. And they're linked actually, the maturation time and the number of cells in newborn linked. So if you have very, very long maturation time, right? So cheaters, what would happen to cheaters? The ones that don't produce, make the FP go zero. They will what? They will take over. In every single culture, there's a chance, some may be early, some late, it will be overrun by cheaters, right? So the maturation time cannot be very long, but also cannot be very short. As we said, we need enough time to see that purple pigment. And the same with, yeah, I still don't understand, so forget it, because my time is almost out. and then the number of cells in newborn as well. If you have a lot of cells in the newborn, then what? Yeah, so like, yeah, then what? If there's a huge number of cells in the newborn, then what? Then each tube would have jitters, right? So they would again look deterministic, like what I said yesterday, the key of the small number of cells, right? And if there's too, if it's too small, you might lose a species, right? So like it's again this. So okay, so I'm done with my session, but I have an announcement to make. So yesterday I was having dinner with some students, and they mentioned that I was telling them about how good the swimming pool is. They said that they regret they don't know how to swim. So I offered to teach them, because I learned it not so long ago. So I still remember the struggle I had by learning swimming. So I offered to teach, whoever interested, right? There's a beautiful swimming pool there. If you're interested in it, it's okay if you don't have swimming suit, just t-shirt. Just basically you have to bring your towel, like a t-shirt, shorts to swim, and a dry set, it's obvious, right? And maybe some soap and shampoo to wash. So we will meet at the gym at 7:20 p.m., right? Because that time it's not crowded anymore. And then for people who want to learn, it's such good exercise, and it's so relaxing. Like after the whole day of this yappa, right? Finally you get to relax. So 7:20, right? Bring all these, and you have to sign in, and you have to change shoes or whatever, right? I don't know the routine, but the point is that it's just a beautiful, beautiful pool, right? So where do you get a towel? I still don't know where do you get a towel.
When you enter, then you will find it there on a chair. They will put it there.
I don't see it, or maybe I was just too quick. I never noticed. Yes, it's provided, right? So you only need to bring basically your and do they have enough? Because I don't know, at least 3 students will show up.
I don't know whether everybody will show up.
but people who know how to swim, right? I mean, then please go to a different time. So don't crowd up the pool, right? So
If you don't know how to swim, you cannot learn in one day.
what did you say? You could, because we're not competing in Olympics, right? So it's fine. It's fine. You only need to know float, right? And then making movements. Even if you go backwards, it's okay. You can exercise, right? It's fine. It's like fine. So that was the struggle I had when I learned swimming, right? So they say kick. So I kicked my legs, I go backwards. The faster I kick, the more backwards I go. The teacher said, why do you not go forward? I said, I don't know why I'm not going forward. So I still remember. But then in the end, it just somehow it just worked. And I don't know if there's a magic, right? But the important thing is just to float and then to make your body move. So, and then you float naturally, right? So it's breathing. I think you should be able to learn, and tomorrow as well. So you have 2 chances of learning that. Thank you very much.